Every living creature on this planet, from the tiniest microbe clinging to a hydrothermal vent to the largest whale crossing an ocean, runs on the same molecular software. Inside virtually every cell, a twisting double helix of DNA carries instructions so precise, so elegantly written that scientists spent decades just learning to read them. For most of human history, that code was fixed. You inherited what you inherited. Evolution moved at its own slow pace. But something remarkable has been happening in laboratories around the world over the past few decades, and the pace of that transformation is accelerating faster than almost anyone predicted. Scientists have learned not just to read the code of life, but to rewrite it. This is synthetic biology, and it may be the most consequential technology of the 21st century. At its heart, synthetic biology merges the logic of engineering with the language of genetics. Where a computer programmer writes lines of code to make a machine do something new, a synthetic biologist writes genetic sequences to make cells do something they never would have done naturally. They design new biological parts, assemble them into circuits, and implant those circuits into living organisms. The result is organisms that behave like programmable machines, capable of producing medicines, detecting diseases, cleaning up pollution, or manufacturing materials that don't exist anywhere in nature. The tools that made this possible didn't appear overnight. For decades, scientists built up a foundational understanding of how DNA works, how genes are switched on and off, how proteins fold and function. Then came a revolution. In the early 2010s, researchers discovered how to harness a bacterial defense system to edit DNA with unprecedented precision. Before these new tools existed, editing a gene was like trying to change one letter in a 3-billion-letter book using blunt scissors and a great deal of luck. Suddenly, researchers could target almost any gene in almost any organism and cut it with surgical accuracy. Broken sequences could be silenced. Faulty instructions could be replaced. Entirely new capabilities could be inserted where nothing of the sort had existed before. The applications that followed were stunning in their range. In medicine, gene editing therapies began moving into clinical trials. Scientists corrected the mutation responsible for sickle cell disease directly inside a patient's bone marrow stem cells. Others targeted cancer-fighting immune cells, engineering them to recognize and destroy tumor cells that the body's own defenses had been unable to stop. The technique proved so versatile that researchers started using it to develop diagnostics, turning molecular machinery into sensors that could detect specific viral sequences and signal an infection in minutes rather than days. Where conventional medicine often treated the symptoms of disease, synthetic biology offered the possibility of addressing the underlying biological instructions that caused it in the first place. Agriculture was transformed as well. Crops were engineered to resist fungal infections, withstand droughts, or produce yields robust enough to feed growing populations without clearing additional land. Scientists used precision editing to silence the enzyme responsible for browning in certain produce, extending shelf life without added preservatives. In other labs, researchers developed grain varieties resistant to the devastation caused by common pathogens. These weren't the crude early-generation modified crops built by randomly inserting foreign genetic material into a host organism. These were careful, targeted changes to a crop's own existing genome, altering individual positions in sequences that had been thoroughly mapped and studied over years of research. But synthetic biology has moved far beyond editing what already exists. One of the most remarkable developments in recent years is the construction of entirely artificial genetic circuits, biological systems designed from scratch to perform specific tasks inside living cells. Scientists have built genetic toggle switches that flip between active and inactive states based on molecular signals in their environment. They have engineered cells that function like logic gates, producing an output only when multiple specific inputs arrive simultaneously. Some researchers have created oscillating circuits that generate rhythmic pulses of protein production, mimicking the biological clocks that govern timing in natural organisms. These genetic circuits are not merely fascinating demonstrations of what is possible. They are foundational to a new kind of medicine. Researchers are developing cells that can be programmed to sense the presence of cancer, evaluate the stage of the disease, and release precisely calibrated therapeutic molecules to trigger the right immune response at the right time. This represents a fundamentally different paradigm from conventional treatment. It is medicine that thinks, responds, and adapts inside the body in real time. Beyond medicine and agriculture, synthetic biology is opening entirely new categories of industry. Companies are engineering microbes to produce sustainable alternatives to petroleum-based plastics. Others are coaxing yeast cells into producing silk proteins far stronger by weight than steel, for use in textiles, medical sutures, and protective applications. Bacteria have been engineered to break down environmental toxins in contaminated soil, remediating industrial pollution at a fraction of the cost of traditional cleanup approaches. Synthetic organisms designed to capture atmospheric carbon and convert it into useful compounds are being tested as potential tools in addressing climate change. Perhaps most astonishing of all is the emergence of synthetic genomes. Researchers have built complete bacterial genomes from scratch using chemically synthesized DNA and inserted them into living cells, which then began reproducing normally. It was the first time a living organism had been brought to life carrying a fully artificial set of genetic instructions. Teams at major research institutions later rebuilt bacterial genomes using a simplified, redundancy-free version of the genetic code. By reassigning the units that specify which amino acids a cell produces, they created organisms resistant to certain viruses and opened the door to producing proteins built from entirely novel, non-natural chemical building blocks that nature never evolved on its own. The convergence of artificial intelligence and synthetic biology is now accelerating everything. Machine learning models trained on vast libraries of genetic and protein data can predict how an engineered genetic circuit will behave inside a cell before a single experiment is performed. Protein design algorithms generate entirely novel molecules with no natural equivalent, folded precisely into desired shapes to perform functions never seen in biology before. What once required years of iterative trial and error in a laboratory can now be narrowed down to weeks of targeted experimentation, guided by computational predictions that are growing more accurate with every passing year. None of this comes without serious questions. The power to rewrite life at the genetic level raises profound ethical concerns that society is only beginning to confront. How do we ensure that these technologies are accessible to people in all parts of the world, and not only to those in wealthy countries or well-funded institutions? Who decides how and when these tools should be applied in human beings? What are the long-term consequences of releasing genetically engineered organisms into wild ecosystems? A gene drive, for example, is a technique that can spread a genetic change through an entire wild population within just a few generations. Used thoughtfully, it could eliminate malaria-carrying insect populations. Used carelessly, it could disrupt ecosystems in ways that are difficult to foresee and nearly impossible to reverse. There is also the biosecurity dimension. The same capabilities that allow researchers to engineer beneficial organisms could theoretically be misused to create harmful ones. As the cost of DNA synthesis continues to fall and the technical barrier to entry drops, the community of people capable of performing advanced genetic engineering grows rapidly. Scientists, ethicists, policymakers, and biosecurity experts are working urgently to establish appropriate safeguards, but the technology is moving faster than the regulatory frameworks designed to govern it. What is clear is that synthetic biology represents a genuine turning point in the relationship between humanity and the living world. For billions of years, life evolved according to the blind pressures of natural selection. Mutations arose at random. Those that helped organisms survive and reproduce spread through populations across generations. Those that did not faded into extinction. It was a slow, undirected, astonishing process that produced every living thing we have ever known or studied. Now, for the first time, an organism has acquired the ability to consciously redirect that process, not just for itself, but for countless other species and for the biological systems on which the entire planet depends. The question is no longer whether we have the tools to reshape life. We clearly do. The deeper question is whether we have the wisdom to use those tools well, to deploy them in ways that ease suffering, increase abundance, and protect the intricate network of living systems that sustains all of us. Synthetic biology has handed humanity an extraordinary set